Okay, for these topics, okay, term as hormonal control during the menstrual cycle. So what are the learning outcome that we're going to learn here? What are learning outcome? First, we're going to look at the role of the meiosis in gametogenesis in both male and female humans. And then after that, we're going to look at the menstrual cycle, human menstrual cycle, by referring to the hormone FSH, LH, estrogen, and progesterone, as well as another hormones that we are going to talk about is the GnRH. Okay, 5 hormones. How these 5 hormones again can help in the female to control the changes in ovary as well as the wall of the uterus during the human's menstrual cycle. And then the last part, we're going to look at the biological basis of the contraceptive pill containing the estrogen as well as progesterone. So we're going to look at how this oral contraceptive pill can help to prevent the ovulation as well as the pregnancy. Okay, another part we're going to put in here, which is already discussed previously in the conservations, where we use assisted reproduction. One of these IVF. So we're going to look at how IVF can be done in more detailed way. Okay, so before we start for this menstrual cycle and contraceptive pill, let us look at the gametogenesis first. And at the same time, we do have a little quick ones for the revision as well, for the meiosis. So gametogenesis is the biological process by which diploid cells undergo the reduction divisions, or we term it as the meiosis, and differentiate into the haploid gametes. Okay, so humans produce the gametes directly through the meiosis from the diploid mother cells, either in the testes for male or ovaries for female. So for male, the way we produce, okay, or the name of the process that we produce the male gamete is termed as spermatogenesis, which is relatively not so complicated compared to the female oogenesis, okay, where female going to form the oocytes or secondary oocyte when they are going to ovulate. Okay, so let us start with the first part, spermatogenesis. And spermatogenesis is the process by which, okay, we start to produce the haploid spermatozoa for the male when the male reach the puberty and develop from who? From the germ cells in the seminiferous tubule. So what is this seminiferous tubule? If you look at the testis of the male testis, then you can see that this is a scrotum. Okay, the scrotum basically means that the bag or the sac. Can you see that? And then inside this bag that hang outside the male body, we do have the structure, okay, like a testis here. Okay, testis, a ball structure. Inside this structure, we can see that we have a lot of this tubular structure inside, termed as seminiferous tubule. And this seminiferous tubule is where the sperm cells will be produced. Are you clear? Okay, seminiferous tubule. Yeah. And then after the formation of the sperm cells were going to be stored inside, okay, will be stored inside this, what we call the epididymis. Okay, epididymis is where the storage of the sperm cells take place. So entire process may take up to about 3 months to prepare, okay, to get the cells matured. Okay, so means that any ejaculations that a male have done now is the sperm cell. The sperm cell are actually from a few months ago, about at least 2 to 3 months ago. Okay. So we look at the spermatogenesis in more detail now. So look at this diagram. Okay, so we have the mother cells. Okay, the sperm mother cells. In this case, we call it as primordial germ cells. Okay, so first thing, primordial germ cell continue to carry out the mitosis. So we have the spermatogonium, singular, and we make many spermatogonia through the process called mitotic divisions or mitosis. So this process actually takes place. You can see that this is the basement membrane. Can you see that? Basement membrane. So those cells that still attached to the basement membrane is termed as spermatogonium. Can you see that? The cell that attached to the membrane is called spermatogonium. So these cells will continue to carry out mitosis to make many spermatogonia. Okay, make many spermatogonia, and these spermatogonia still attached to the membrane. Are you clear? So at this moment, they are diploid, 2n equal to 46. At this moment, they still diploid. Okay, but once this spermatogonia detach from the basement membrane, then they were developed into what we call as a primary spermatocyte and start the process of meiosis. So it means that, can I say that they detach from the basement membrane? When they detach from the basement membrane, then they're going to form. Can you see that? They still attached, right? The basement membrane. When they detach from the basement membrane, they will enter into the prophase 1 of the meiosis and continue to divide. Okay. So after the first divisions, we do know that meiosis 1 is to segregate the homologous chromosome. Therefore, when the cells ends the meiosis 1, they will enter into the second meiotic division. So in the second meiotic divisions, then we term them as a secondary spermatocyte. Are you clear? We term them as a secondary spermatocyte. At this moment, they become haploid already. So write down spermatogonium 2n, primary spermatocyte 2n, but when secondary spermatocyte is n. But for each of the chromosome, they still have 2 sister chromatids. So therefore, secondary spermatocyte have to complete the second meiotic divisions to form spermatid, which is equal to n equal 23. But at this moment, can you see that they still inside? Okay, can you see that they still haven't formed or grow into this fully matured sperm cell with the tail? Are you clear? Can you see that? So at this moment, we term them as a spermatids. So they were matured into the sperm cells. Are you clear? They were matured into the sperm cells, okay, in the process called spermiogenesis. Are you clear? Terms, okay, form sperm cells. But the sperm cells at this moment, can you see that? They still link with the Sertoli cells. They still link at or attached to the wall of this seminiferous tubule. But when we release them, so the spermiation process take place, means that they are fully matured. Okay, so few process that we need to know here, guys. First, you will see that we do have the spermatogonia. Okay, spermatogonia attached to the basement membrane. Okay, spermatogonia attached to the basement membrane. They continue to carry out mitotic division to form many more spermatogonia. Okay, form many spermatogonia. Okay, so when this spermatogonia detach from the basement membrane, then they start or initiate the first meiotic division. So at this moment, they are still 2n. So they are termed as the primary spermatocyte. They enter into the first meiotic division, start with the prophase 1. Okay, and then they continue from prophase 1, enter into metaphase 1, and then anaphase 1, telophase 1, and then this cytokinesis take place. They form 2 secondary spermatocytes, and at this moment they are haploids already. So once they finish the second meiotic divisions, they form 4 spermatids. Okay, 4 spermatids which are haploids, but spermatid is still attached to the wall of the seminiferous tubules, and from there they start to grow. Okay, they start to grow and develop into the sperm cells through the process called spermiogenesis. Okay, to form the sperm cells. Once they detach, okay, and freely move in the lumens, then spermiations take place where the sperm cell, which is haploids or which are haploids, now fully develop or fully matured. Okay, they can actually move out from the seminiferous tubule and enter into the epididymis. Okay, so look at the process here. Okay, so first, okay, each human spermatogonium, or called germ stem cells, give rise to many spermatogonia by the process of mitosis. Therefore, the number of chromosomes in each spermatogonium cell is conserved at 2n equal to 4. So this is the first process. Number two, second process, some of these spermatogonia lose the contact with the basement membrane and differentiate into 2n diploid primary spermatocyte or grow into this primary spermatocyte. Okay, and then at the end of the first meiosis once, they form two secondary haploid spermatocytes already. Okay. And then at the end of the meiosis 2, they're going to form 4 haploid spermatids. Okay, 4 haploid spermatids. And the spermiogenesis result in the development of the haploid spermatids into the sperm cells. And sperm cells are released from the connections to the Sertoli cells or the wall of the seminiferous tubule through the process called spermiation. Okay, so let me actually summarize for you guys. So gonium. So spermatogonium is 2n. So carry out mitosis to form many spermatogonia. Okay, so some spermatogonia actually detach. Okay, detach from some of these spermatogonia detach from the basement membrane. Then they develop into the primary spermatocyte. Okay, so one of them. Okay, a few of them. Okay, so again, this 2n primary spermatocytes 2n. So this primary spermatocytes continue to finish the meiosis 1. Then they develop into the secondary spermatocyte. Okay, how many of them? So from one primary spermatocyte, we're going to get 2 secondary spermatocyte. So now it's n. So they continue to finish the meiosis 2. Okay, forming what? Spermatids, which is also n. So from 2, you get 4 spermatids. Okay, so spermatids develop into the sperm cells, which is n. Okay, by the process of spermiogenesis. Means that develops sperm cells. Are you clear? So still 4. So sperm cell will be released from the Sertoli cells, and becomes, a fully developed, or at least, develops sperm cells through the process of spermiation. Okay, so this is the summary. So you can take a photo you need. Okay. So this is the summary of the spermatogenesis. Relatively simple and non-complicated. Relatively simple and non-complicated if you compare to the female later. Okay. Any questions so far, guys? If no, then I'll continue. You have notes. So you look at the structure here. This is what we say that once seminiferous tubules. Can you see that? This is a wall of the seminiferous tubule. So this is where the lumens of the seminiferous tubule. So the cells that attach to the basement membranes, they are spermatogonia, and you can see that the cells. Can you see that? So those are the either primary spermatocyte or secondary spermatocytes. Okay, and then you can see the spermatids and the sperm cells. Okay, so how to differentiate? Question sometimes will ask you how to differentiate the spermatogonia. So how to know? To search those that attach to the basement membrane. Those are the spermatogonia. Okay, so what's the Sertoli cells? So Sertoli cells, they are the ring of the special nurse cells which present at the wall of the seminiferous tubules. So the fluid that secretes by these Sertoli cells, we're going to stimulate the spermatids to become the sperm cells. Sertoli cells will be the one that secrete the fluid which stimulates the spermatids to become the sperm cells or develop into the sperm cells. And if you look at the structure here, okay, so sperm cell will have the head and have the midpiece or middle piece with the tail. So where is the mitochondria? This is where the mitochondria is. So still remember previously we talked about when the fertilization takes place, we talked about mitochondria if and the molecular clocks hypothesis. We say that mitochondria only can be inherited from the mother or maternal lines, correct or not? Why? Because the head, again, the head is the one that penetrate into the secondary oocytes, okay, and then release the nucleus, but not the middle piece. But current research actually showed that, yes, there is a possible of the mitochondria from the sperm, okay, enter into the secondary oocyte. So one structure here is very, very important, is known as the acrosome, okay, known as acrosome. Acrosome contain the enzymes, okay, that to facilitates the penetrations of the sperm cells during the fertilizations. Okay, so this is the under light microscope. You can see the head and also the middle piece and also the tail of the sperm cells. So generally, testes produce about 200 to 300 millions of the spermatozoa or sperm cells daily, but to become and to survive one is actually 1/3 of them only. Okay, or half of them become or survive. So the location of testes is specifically important as the process of spermatogenesis requires a lower temperature to produce a viable sperm, generally 1 to 8 degrees Celsius lower than the normal body temperature. Are you clear? So that's why testes, okay, in terms of evolution, it hangs out, okay, it's hang outside of the body, okay, for the male rather than keep it inside. Clear? So from the seminiferous tubules, the cell will travel to the rectal testis. This will act as to concentrate the sperm cell by removing the excess fluid. Then the cell will move to epididymis, where the sperm is stored and undergo the final stage of the maturations. So for humans, the entire process of spermatogenesis varies, estimating about 70 days or 2 months, or it may take up to, including the transport and ductal system, it may take up to 3 months. Okay, so means that if you want the sperm cells to be active and healthy, so you may want to produce a new sperm cells. Okay, let's say for example you quit the smoking. Okay, for example you quit the smoking, and to quit the smoking, the effect may take about 3 months. It won't immediately. Okay, so we do have these two processes which are not in our syllabus, but come out before. One is called as a capacitation, another one is called acrosome reactions or acrosomal reactions. So following ejaculations, sperm cell leave the male body and enter into the female reproductive tracts. Okay, so during the sexual intercourse, the sperm cell will enter into the vagina, swim across or move across the cervix, enter the uterus, and move all the way to the fallopian tube or oviduct, where the fertilization takes place. Okay, and during this process, when they're nearer approaching this secondary oocyte, then the sperm cell will carry out these sperm capacitations, where the removal of the cholesterol and also the glycoprotein from the head of the sperm cells to destabilize the acrosomal head and also give a greater mobility in the sperm cells. So the sperm cells were going to destabilize the membrane so that later we can have the acrosome reactions. So as the sperm cell approaches the zona pellucida of the egg cell, so egg cell actually contain layer of the jelly coats, okay, which is known as zona pellucida, surrounding the acrosome, fused with the plasma membrane of sperm head, exposing the contents of the acrosome. So this content include the surface antigen necessary for the binding of the egg, okay, cell's membranes, as also the enzyme which is responsible for breaking through the egg tough coating and allowing fertilization to take place. So for you guys, you just need to know the name. There are the process known as sperm capacitation and acrosome reaction, but you do not need to know the detail. No question will ask, okay, describe the process of the sperm capacitation and also the acrosomal reactions. Okay, so generally this diagram, you can see that this is the fresh sperm cell. Okay, sorry. This is a fresh sperm cell. So what this fresh sperm cell actually did? This fresh sperm cell, when approached to the oocyte, can you see that the oocyte contains a layer known as the zona pellucida? Can you see that? Zona pellucida. So this layer, if you want to break this layer, you need to have the enzymes to release the enzyme. So first thing, you can see that capacitation take place. When the capacitation take place, it makes the sperm cell actually more active so that they can swim. They provide a more what we call the force for the sperm cell actually to enter and to break this zona pellucida. So in order to break this zona pellucida, then acrosome reaction actually take place. So capacitated sperm cells will undergo the acrosome activities, and then you can see that the cells now enter into this zona pellucida attached to the what we call this the surface antigen, allowing the fertilization to take place. So do you realize that the middle piece never enter into the oocyte? Why? Because when this acrosome activity reaction take place, the nucleus will be the one that enter into the oocyte, okay, or secondary oocyte. Okay, so with this, I have done for the spermatogenesis.